Hello, hello, honors biology students. This is your lab on death in the rainforest. This is a forensic case study on blood, food, plants, plankton, bacteria, and viruses that are going to help you determine how 10 UCLA professional cancer researchers died in the Brazilian rainforest. So let's get excited. So let's go and talk about what happened. All right. So do me a favor and think for a second. How many of you know someone who has or had cancer? There's a lot of you. There's a lot of us. And so in 2017, it was estimated that there were over 100 million people around the world that had some type of cancer. In 2018, it was estimated that over 1 million new cases of cancer would be diagnosed in the United States alone, and over 600,000 people would actually die from the disease. And this is from the National Cancer Institute. So it's imperative that we in 2020 find a cure to cancer. Okay, now there's various research that's going on around the world to help stop cancer, and it's ranging from cures in organic foods to raw food diet in fighting cancer to even immunotoxins. Immunotoxins are toxins that actually attack the immune system. So yesterday a group of 10 experienced field researchers from UCLA died in the tropical forests of Colombia. So that's in Brazil, while conducting an investigation on medical alternatives. So instead of having chemotherapy and taking all these, chemical derived pharmaceuticals to help treat cancer, they were actually looking for a cure derived from both plants and animals. And this is based on the evidence that you guys are going to find today in today's lab. You're going to decipher how this group actually ended up dying after just 3 hours of exposure in the forest. Okay, so that's key. So 10 researchers ended up dying in Brazil after 3 hours of exposure in the Brazilian rainforest. Now, what were they actually investigating? So these here are our main topics. We're going to be looking at organic foods such as onions and potatoes. We're going to be looking at plants, whether it's freshwater alodia, which is a freshwater plant, or marine phytoplankton. We're also going to be looking at toxic animals and toxic plankton such as red tides. We're going to look at bacteria as well as viruses. Okay, so here's what you guys are going to be doing. You're going to be exploring all this information that I'm going to be giving to you. You're also going to be stopping the video along the way, so stop it as many times as you want. And you will also be using the lab handout that you downloaded for this video. And after you see a slide, stop the video and go onto Google or use your phone and research some of the questions that correspond to each of these slides. So throughout the study, just keep in mind what items you think might have caused these researchers to die in the rainforest, and items that you definitely think that did not cause it. Okay, so the first items here are your food. So this here is an onion and a potato. I've already pre-labeled them for you, so all you have to do right now is stop the slide or stop the lecture and draw out what you see and go ahead and label it in the spaces provided on your handout. When you are done labeling, please go ahead and take out your phone or the internet and Google questions in the lab section. So what is a leucoplast? What are chloroplasts? And of course, could onions and potatoes be toxic to humans? So do me a favor, stop the video. All right, Next section here, guys, we're going to look at plants. So again, we're going to determine if plants can actually kill people. And you guys know from red tides that that's definitely possible. So the first slide here, you're going to be drawing out Elodia. Elodia is a marine plant that grows in fresh water. And again, the Brazilian rainforest is filled with fresh water and brackish rivers. You also are going to stop the video and draw diatoms here on the right. Diatoms are a marine plant that fills our ocean, that helps with pretty much like 90% or 50% of the air that we breathe comes from the photosynthesis from these diatoms. Okay, so again, stop the video, draw it out, label what you can, and then remember to Google and or use your textbook in researching the questions that are provided on your handout. Okay, next page we have our animals. You have a human blood prepared slide. Again, these are all labeled for you, so make sure to stop the video and write that out. And I want you guys to correlate human blood, which have both red and white blood cells, to a frog's blood. So use this data to help you guys analyze what the frog's blood is. Okay, and again, make sure to stop the video and then make sure to also Google the questions that are provided in the lab. Here's our next slide. This is your zooplankton or zooplankton. These are marine animals. Zooplankton are actually like baby larvae. So baby crab, baby shrimp. It could also be organisms that really never grow up to be any bigger, like, you know, krill. And so what I want you guys to do is to Google, so again, draw just one or two of these. You don't need to draw all of them, but draw one or two, and then please go ahead and Google toxic dinoflagellates. So some of these can actually create red tides as well, even though they're an animal. If they have a bunch of nutrients in the ocean, they will reproduce like crazy, and all of a sudden they also will turn the color of the water red, brown, you know, green, or even yellow, depending on the pigments that they actually have in their body. Now again, these are animal organisms that can also cause red tides, and they can also be extremely toxic. So take a second, stop the slide, and then make sure to Google toxic dinoflagellates or toxic zooplankton. Okay guys, next slide we're going to be looking at is prokaryotes. Pro means before, karyo means nucleus. So this is before a true nucleus. Eukaryotes, eu in Latin means true, karyo means nucleus. So this means that we have a true nucleus. So there's three types of bacteria here. There's bacilli, which is more of a bar shape. Cocci are more of circles, and then Sporilia are these like kind of like spiral shaped bacteria. So here is a microscopic slide of pneumonia. So if you guys, you know, this is pretty much as you would see on your school microscope. So if you would zoom in even more, pneumonia is actually a cocci bacteria. So it's a bunch of little circles all, you know, kind of put together. That's why pneumonia is called pneumococcus, right? Again, make sure you guys Google toxic bacteria, where it's found, maybe effects on it, so that we can see maybe if these researchers died from, you know, a toxic bacteria, or maybe was it a toxic, you know, red tide. And then we're going to correlate that to eukaryotes. Okay, so these, if I were to have you guys in the class, you would take a toothpick, you would scrape, gently scrape the inside of your cheek cell, would add a little bit of methylene blue to highlight this nucleus. And this is actually what you guys would see. So you'd see these nice big kind of roundish cells. Inside every single cell of your cheek are these nice big blue dots, and that's your nucleus. So if we were to take a needle and go inside the nucleus, I could actually extract your DNA and make a clone of you. Okay. Now, in comparison, if you guys see how small this pneumonia is, these little tiny blue dots are bacteria compared to our big single cells, right? So again, make sure to stop the video, draw out the bacteria and your cheek cells, and then make sure to either use your textbook or Google to answer some of these questions. Okay. Next slide we have is the virus slide. I actually changed the last video, so on your handout, this one's actually new, but you're going to stop the slide. Again, make sure to draw out what the Zika virus is, and then let's go ahead and look at two videos here that talk about the Zika virus. So here's one. Okay, so heading on back again, there are two videos here that you can actually, kind of look up if you want more information about Zika. Please make sure when you are done with this lab, you're going to write out three things that you definitely believe are not part of the death cause. Okay, so these are the three things that I definitely don't believe caused the researchers to die, and then I want you to figure out, okay, well, what do you think actually caused 10 UCLA researchers to die in just 3 hours? So write out your claim on your handout and make sure use evidence from the lab that we've just done. Okay, now in class, we're going to go through the rest of this. So you guys don't need to pre-fill this out. In class next time, we're going to be doing a class discussion. So again, make sure you guys have a claim. We're going to be talking about your claims. We're going to be doing maybe a little bit further research as far as looking into more specifically, toxic plants and toxic red tides. And then we're also going to be doing a quick activity on Jamboard where we're going to be connecting all of this knowledge that you guys just did, and we're going to end this activity with a quick modeling assessment on perhaps what you think got into these researchers, how they got into them, and then perhaps how they actually affected the cells. So this is where we're going, okay? Feel free to rewatch this video as many times as you want. Again, our focus is on what organelles perhaps interacted with this toxin and or food or anything that we've been researching, plants, animals, virus, bacteria, that may have killed 10 researchers in about 3 hours after they got into the rainforest. All right, guys, I will see you back in class. This is Holland Chambers, checking out.